Okay, so the person you're looking at right now is Thomas Jefferson. Let's have a bit of historical chismes before we go to the actual lecture. I'm sure you know that Thomas Jefferson is the third US president. Now, before becoming the US president, he was one of the authors of the Declaration of Independence, and he was also the Secretary of State and one of the founding fathers of the University of Virginia. So he's rather well regarded in US history. But even with that stature, he does have a lot of skeletons in his closet. Ironically, his family owned more than 100 slaves, but he does have one favorite. Her name is Sally Hemings. Well, Sally Hemings is not a stranger to the Jefferson household. As a matter of fact, she is the black half sister of the First Lady. But she is not considered a family because, well, she is colored. When the First Lady died, Sally Hemings was about 14 years old, and she was given the task of taking care of the Jefferson children. So siya yung nanny. When she turned 16, she started giving birth to eventually a total of six children, all of whom were rumored to be fathered by Thomas Jefferson. Of course, Thomas Jefferson never admitted this, but the one of the children, the youngest of whom is Eston Hemings, um, his current day descendants claim that Thomas Jefferson is their ancestor, despite the lack of any historical evidence to prove such claim. So, with the development of molecular DNA technology in 1998, DNA test was used to settle this historical dispute and allegation. So we're going to take a look at the result later on, and you are going to interpret the result and make your own conclusion. But of course, before you can do that, we are going to study first the introduction to molecular medicine so you have the necessary tools to understand and interpret the result. What is molecular medicine anyways? So this is the application of the techniques, the technology, the principles, the methodology that are used in molecular biology, biotechnology, and molecular genetics to the field of medicine. What for? To understand human health so it can be used for research, for diagnosis, and to a limited degree for treatment of some diseases. So it aims to understand how health is maintained, as well as to research on the origins and mechanism of the diseases. Okay, so what are your what are the topics that will be covered here? So we'll discuss first restriction endonucleases, followed by the discussion of recombinant DNA. And then we'll proceed with how DNA can be propagated, starting with a cell-based um, technology for propagating DNA, followed by an in vitro process called PCR. Then we'll discuss the different applications of PCR, including examples. Then we'll continue with DNA sequencing, the different blocking techniques, and then we'll end the lecture with DNA microarray. So what is expected of you after studying this topic? So you only have three learning objectives. The first is that you should be able to explain the principles of molecular DNA technology, the ones that will be presented today. You should be able to discuss their applications as well as limitations in the field of medicine. And most importantly, you should be able to analyze the data that are obtained from molecular DNA procedure. And this is how you shall be mainly tested on during your major examination. So there will be a lot of cases, a lot of results, and I will be asking you to interpret and give your findings in the examination. So let's start first with restriction endonucleases. Now, we always start the discussion with restriction endonuclease because molecular biology became possible with the discovery of these enzymes. So these enzymes are derived from microorganisms, and like all other enzymes, they are highly specific. What is their action? Since there are endonucleases, their action is to cleave DNA. But they don't just cleave DNA sequences randomly, but rather they are restricted to certain specific DNA sequences. And the DNA sequences that they act on are those that are said to be palindromes. What is a palindrome anyway? In the languages, a palindrome is a word or a phrase that is the same when read forward and backward, like Dennis sinned. Try reading it backward, it's still the same. I have here a lot of examples of palindromes, such as was it a car I saw or a cat I saw? No lemons, no melon. All of these are palindromes. Sa Tagalog, marami rin tayong palindrome, katulad ng nasa bayabasan. Pag binasa mo yan pabaliktad, ang kalalabasan ay nasa bayabasan pa rin. Now let's apply this concept to a DNA sequence. The classic example of a palindrome is GATC. You might be wondering, why is this a palindrome? Now remember that DNA sequence should always be read in the 5' to 3' direction. So the top strand is read as GATC. The bottom complementary strand still read as GATC, the same exact sequence as its complementary strand on the top. So this is a classic example of a palindrome. We have here a tabulation of the common restriction endonucleases, the microorganisms where they come from, and the specific palindrome sequence they recognize and cut. Let's take a look at EcoR1. If you notice, the names of the enzymes are derived from the microorganisms where they come from, like EcoR1. We take the first letter of the genus, followed by letters coming from the species. And this particular restriction endonuclease recognizes and cuts GAATTC. Thus, when presented with the DNA sequence, it will scan the DNA and look for this. Once found, it will cut that DNA at this particular sequence. If you give it a DNA and it has all the other sequences but none of this, then dead ma. Wala siyang gagawin sa DNA. It will never cut that DNA. Whenever restriction endonuclease cut a DNA fragment, it always produces 3 possible outcomes. The first is shown here, as exemplified by SMA1. In this example, it cuts exactly at the very center of the palindrome sequence it recognizes. This is referred to as a symmetric cut. Symmetric cut. The resulting DNA fragment would have very clean edges, referred to as having blunt ends. The second possible outcome is demonstrated here, exemplified by BAM-H1. Rather than cutting exactly at the midline, it cuts off center of the palindrome sequence. This is referred to as an asymmetric cut. The resulting DNA fragment would have a 5' overhang. So pag pinutol mo ganyan at pinaghiwalay mo, may 5' overhang here, as well as a 5' overhang at the top. Sa Tagalog, ito yung tinatawag na lawit. Now, these 5' overhangs have the tendency to reanneal with another DNA sequence it is complementary with. So thus, this 5' overhang are said to have sticky ends. They can piece together with another complementary DNA. So this is the kind of restriction endonuclease that can be very useful if you wish to piece together DNA from different sources. In layman, this is called genetic engineering. Well, the third possible outcome is exemplified by this enzyme. So just like the second one, it also cuts off center. So it's also an asymmetric cut. But rather than producing a 5' overhang, what is produced here is a 3' overhang. Well, nonetheless, the ends are still said to be very sticky. Like what I said earlier, the birth of molecular biology became possible because of the discovery of restriction nucleases, and it gave rise to several technologies, such as the production of recombinant DNA. What is a recombinant DNA? This would be DNA that are pieced together, and this DNA comes from different sources. For example, we have here a closed circular plasmid that comes from a microorganism. And let's say this is a DNA from a higher organism like humans. So this plasmid contains GAATT sequence that can be cut using EcoR1, while this foreign DNA can also be cut using the same restriction endonuclease if it also possesses the same restriction enzyme cutting site at the same palindrome sequence. Thus, when exposed to that enzyme, they will produce sticky ends that are complementary to each other. So when mixed together, those two DNA fragments are expected to reanneal, producing now the recombinant DNA. As you can see here, the gray part comes from the microorganism, while the violet part comes from another organism. Now, at this point, we only have few copies of this recombinant DNA because the production of recombinant DNA is really not as easy as this simplified diagram. So our next goal is to replicate or reproduce more of this recombinant DNA. This is where DNA propagation will come in. I'll be presenting two methods for DNA propagation. The first is cell-based, where we shall be utilizing a host cell that contains replication machinery in the form of DNA polymerase, among other enzymes. That recombinant DNA will be introduced into that host cell to allow that recombinant DNA to use those enzymatic machinery for the process of replication. The second one, PCR, is an in vitro process. Let's start first with cell-based. Cell-based propagation of DNA can also be referred to as cloning. So cloning, in its simple definition, is really just the production of large quantities of identical DNA molecules. But cloning can come at different levels. The most basic is simply the reproduction of identical DNA. And the more advanced form of cloning is the cloning of an entire organism, like Dolly. So since this is just an introduction, we'll just stick to the most basic of all cloning, which is the production of large quantities of identical DNA molecules. So here we need a host cell, and we need to introduce that DNA into that host cell. For us to be able to introduce the DNA we wish to replicate within the host cell, we need a carrier in the form of a vector. So there are different classes of vector, such as the plasmid, the lambda, and the yeast artificial chromosome. Lambda and yeast artificial chromosome are the more sophisticated kinds of vectors, which are beyond the scope of what we can discuss in this introduction to our topic. So instead, we shall focus on the most basic of all the vectors, which is the plasmid vector. So plasmid vector, like what I said, is the most basic of all vectors. This is actually a piece of closed circular DNA found in microorganisms, and this is distinct from the bacterial genome. For a plasmid to be useful as a vector, it should contain three DNA sequences. The first is it should contain an ORIC question. What does ORIC stand for? Based on our lecture last week?
Origin of
Anyone?
replication.
Very good. Now it should also contain a replication S or a restriction endonuclease cutting site. In other words, it should contain a palindrome sequence that can be recognized by a restriction endonuclease. And the third enzyme, the third sequence is an antibiotic resistance gene whose significance we shall be discussing in a little while. there's a limit to the size of DNA that can be carried by a vector. It should be less than 10,000 base pair in length. That is rather short if we're going to compare it in the grander scheme of things. So if you need to clone or propagate larger segments of DNA, you need to use the more sophisticated vectors like the lambda, the yeast artificial chromosome, or the very sophisticated retroviral vector. Okay, so let's start now with the process. This type of propagation is divided into four major steps. The first event is the production of recombinant DNA, followed by introduction of that recombinant DNA into the host cell. We refer to that process as transformation, followed by selection, and the final stage is propagation and purification. So let's start from the very beginning, the production of recombinant DNA. So say, for example, highlighted in violet is the DNA sequence we wish to propagate. That is our sequence that we wish to, well, replicate. So we are going to cut the DNA with the appropriate restriction endonuclease, EcoR1 in this case. Our vector is shown here. It's a plasmid. It should contain three DNA sequences. The first is an origin of replication, not shown in this example. The second one is an antibiotic resistance gene. And the third one is a palindrome sequence that can be recognized by EcoR1. So thus, when digested with EcoR1, it would have opened up, it has sticky ends, and it is ready to accept this DNA fragment. So when mixed together, they will reanneal, forming now that recombinant plasmid DNA. Now we shall introduce this DNA into a host microorganism. The usual host microorganism is an E. coli previously treated to render the cell membrane permeable. Now, some microorganisms are successful in being transformed in that they are able to take in the plasmid, just like students in a classroom who are able to imbibe the lesson of the lecture. And then there are microorganisms that fail to take in the plasmid, like students who fail to take in the lessons of the lecture. So in the end, we are only interested in the students who have learned something, and we wish to eliminate the students who have not learned anything. So in the classroom, how do we do that? How do we separate the students who have learned something from the students who have not learned anything? We subjects the students to letter T. And what is that T? Torture. Okay, sorry, test. But essentially, they're the same thing. So just the same, these microorganisms will also be subjected to a test. They will be placed in a hostile media, a media that contains antibiotic. These microorganisms, do you think they will survive in this media? Yes or no? Wale, wala nakikinig. Hindi nyo alam sagot. Yes or no?
No po.
No, because they do not have the antibiotic resistance gene. On the other hand, these microorganisms definitely will survive because they do contain the antibiotic resistance gene. So in the end, only the microorganisms that were successfully transformed will survive. Now, since this plasmid contain the origin of replication, they can use the machinery within the host cell, such as the helicase, SSB, the DNA polymerase, to so that the plasmid can undergo the process of replication. So every time the microorganism multiplies, along with its multiplication, the plasmid is also replicated. So the final stage is simply isolation and purification of that recombinant DNA plasmid. So you might be wondering, what shall we do now with that propagated recombinant DNA? There's a lot of things that can be done with recombinant DNA. One of them is to establish a DNA library. There are two kinds of DNA library, a genomic library and an mRNA library. Now, since we are very limited with time, I will not be discussing that anymore. Instead, you shall be responsible for it. Read on that DNA library during your individual study. The other things that can be done to a recombinant DNA is that it can be expressed into a recombinant protein, or we can reintroduce that recombinant DNA into the genome of another organism. Let's discuss this last two. That recombinant DNA can be expressed into a recombinant protein. Now we need a second kind of vector for this one. This is called an expression vector. What makes this unique from the first one is that it should contain a gene that can be expressed under certain conditions, like in the presence of lactose. So our favorite is an expression vector that contains lac operon, because as you know, in the presence of lactone, that lac operon is transcribed and translated into the protein beta-galactosidase or lactase. We shall be exploiting this property. The LAC-Z gene is taken out by restriction enzyme digestion, and in its place, we place there the DNA that we have previously isolated and propagated. Grow this in a media that contains lactose. That gene that we have previously isolated and propagated is now expressed into the desired protein. How useful is this? In the olden times, the insulin that was utilized back in the 80s was derived from the pancreas of cows and pigs. They work fine, but the amino acid sequence is not exactly the same as that of human insulin. Thus, they were very antigenic with repeated use. Nowadays, the insulin that we use has the same amino acid sequence as human insulin. It doesn't mean they were extracted from the pancreas of cadavers, but rather the gene for insulin was isolated, propagated, and inserted into an expression vector similar to this. Another thing that can be done with a propagated recombinant DNA is that it can be reintroduced into the genome of another organism. In this example, green fluorescent protein gene that is originally found in jellyfish was isolated and propagated. Then that green fluorescent protein gene was transferred into the genome of the mice during its embryonic stage. Therefore, when it developed into a full-grown organism, each and every cell of its body contains that gene which is expressed into a protein, which of course fluoresces with exposure to UV light. You might be wondering, what is the significance of this particular experiment? Well, it turns out that it's a proof that if you have a non-functional gene, or if you have a missing gene, it can actually be replaced with a functional one. And that is the basis for gene therapy. In this example, the patient has, say, beta thalassemia, where the beta globin gene is defective. It can be cured from it using this gene therapy. Stem cells from the person's bone marrow are isolated. The red helix here represents the defective beta globin gene, which can be replaced, not repressed, replaced. So we need to isolate DNA from another source, package into a very sophisticated vector. This is a retroviral vector that has the capability to insert that beta globin gene into the stem cell. When successfully inserted, this stem cell is now capable of producing beta globin gene. Thus, they are reintroduced back into the bone marrow of the patient. This eventually not just manages, this eventually cures the patient of beta thalassemia. This is a simple video animation that illustrates what we have discussed so far. So this test tube now contains the plasmids that will act as the vector. So it contains the origin of replication, the antibiotic resistance gene, and a palindrome sequence. In this case, it's GAA-TTC, which can be recognized simply by echo R1. Now, the piece of foreign DNA we wish to replicate or propagate was previously caught using the same restriction endonuclease. Thus, it has complementary sticky ends. So when mixed with the open plasmid, they are going to reanneal together, producing now the recombinant DNA plasmid. Now we need to propagate this recombinant DNA. This is where the microorganism will come in. The usual microorganism for this is E. coli. E. coli was previously treated to render the cell membrane permeable. So some microorganisms are able to successfully take up the plasmid, so they have been transformed. And then there are microorganisms which were not transformed. So we wish to eliminate those microorganisms. This can be done through the selection process. These microorganisms will be transferred to a hostile media. It contains an antibiotic. The usual antibiotic is ampicillin. So, of course, in this media, the microorganisms that were transformed contain the antibiotic resistance gene, and they are expected to survive, while those that were not transformed eventually perished. Let's go to the plasmid. Since it has the origin of replication, pwede siyang makigamit ng helicase ng SSB and DNA polymerase within the host cell. So it can undergo the process of replication. So every time the microorganism undergoes binary fission, a copy of that recombinant DNA plasmid is segregated equally within the host daughter cell. Imagine this happening trillions and trillions of times. We now have propagated the recombinant DNA. All right, so we're finished now with cell-based propagation of DNA. Let's go to the next method, an in vitro process called PCR. This is described as an in vitro amplification of DNA. So in essence, we are simulating the DNA replication process that we have discussed a few weeks ago, but on a test tube. So we can describe PCR as a method of amplifying a target sequence of DNA. The maximum length of DNA is 600 base pair. So this is really short compared to the 10,000 base pair that can be propagated in cell-based cloning. 600 base pair is equivalent to 200 trinucleotide repeats. So just remember that. So since this is simulating the replication process in vitro and carried out at a temperature that oscillates and sometimes reaches near boiling point, it will require a DNA polymerase that is stable at this very high temperature. We call this DNA polymerase Taq polymerase. It's an in vitro process simulating replication. And conventional PCR is composed of 30 cycles. Each cycle is made up of 3 steps. After the 30 cycles are over, we have generated more than 1 billion copies of DNA. This is enough copy for us to actually visualize them with our naked eye. Nakikita na sila sa dami ng DNA na na-produce. So what are the 3 steps in PCR? DNA denaturation, DNA primer annealing, and primer extension by Taq polymerase. If it sounds familiar, because really what you're doing here is you are simulating the DNA replication process we have discussed last week. So let's illustrate those 3 steps. So let's say this is the parental DNA strands. Highlighted in violet is the piece of the segment of DNA we wish to amplify or replicate or multiply. The first step is to separate those 2 parental strands from each other. In the replication process we have studied last week, this was accomplished by the enzyme helicase. But in this process, since this is in vitro, we can simply raise the temperature to near boiling point, 92, 94, or 96 degrees Celsius. Then the temperature is lowered to about 40 degrees Celsius to allow the binding of preformed primers. These primers serve the same purpose as the primers we have studied last week. But what makes them different? Well, number one, they are preformed rather than synthesized by the enzyme primase. Number two, they are artificial. Number three, they are composed of DNA rather than RNA. After the primers bind to the flanking sequence of the target region, the temperature is raised to about 70 degrees Celsius to activate now the Taq polymerase. That will now synthesize the DNA in the 5' to 3' direction. So at the end of the first cycle, there's now 2 copies of the DNA. So let's recap. In the first cycle, the steps include denaturation, primer annealing, and primer extension. 2 copies of the DNA have now been produced at the end of the first cycle. Those 3 steps will be repeated over and over again. In the second cycle, after the second cycle rather, we now end up with 4 copies. And at the end of the third cycle, 8 copies are produced. So the amplification process is exponential. This is the reason why at the end of the 30 cycles, more than 1 billion copies of the DNA have been produced. So I have a question before we continue. So I'm not going to, this is not recitation, I'm just curious. Any one of you has experience with PCR? Meron ba?
Yes po.
Okay, what's your name?
Legaspi po.
Legaspi. So how long does, ano yun, conventional or RT-PCR?
So conventional po to.
Okay, what's your PMED?
Bio po to.
Okay, so how long does it take to complete that 30 cycles?
Usually po, Dok, 1 to 2 hours po namin siya ginagawa.
So isang sini yan, 2 hours. Okay, how do you confirm that you are successful in your amplification process?
Nag-agarose gel electrophoresis po.
Okay, so what are you going to check in the agarose gel? Two
Yung
things.
amplicons.
Okay, so how do you, the presence of a band representing the amplicon, and number two,
Yung
the size, di
size.
ba? Okay. So this is a simple video animation that illustrates PCR. So this is an in vitro process. So on the test tube, it contains the materials needed for PCR. We have there the genomic DNA, and we're zooming in into the target sequence we need to amplify. So highlighted in green is the DNA sequence we're going to amplify. So as always, the first step in the first cycle is denaturation to separate the two parental DNA strands. Temperature is lowered to allow primer annealing. Temperature is raised to about 70 degrees Celsius to allow primer extension by the enzyme Pac polymerase. Through polymerization of the dNTPs. So that's the end of the first cycle, 2 copies. Second cycle, repeat. Denaturation, followed by primer annealing, and then primer extension. Four copies are produced. Now take note that those amplicons or DNA fragments are not of uniform size yet. But by the end of the third cycle, we are now going to see the appearance of DNA fragments or amplicons that are of uniform length, like here. So in the fourth cycle, we see the continuous rise of the DNA fragments that are of uniform size. Now, by the end of cycle 5, the DNA fragments or amplicons that are of uniform size outnumber those fragments that are of varying lengths. So let's fast forward to cycle 30 because we don't want to stay here for 3 hours. At the end of the cycle 30, more than 1 billion copies of DNA fragments of uniform lengths have been produced. Like what we had mentioned earlier, to confirm successful amplification, these DNA fragments or amplicons must be subjected to gel electrophoresis. We check for the presence of a band indicating successful amplification, as well as check for the size of the DNA fragment to check if we have successfully amplified the correct DNA sequence. right? So you might be wondering, what shall we do now with PCR? It does have a lot of medical applications. As a matter of fact, PCR is so important that we can divide molecular biology into two eras, the pre-PCR era and the post-PCR era. So PCR is virtually the workhorse now for molecular biology. So we can use PCR to investigate a potentially defective DNA sequence. So it can be used for research as well as for diagnosis. Now, for diagnosis of disease, we're limited to diagnosing monogenic disorder. So we cannot use PCR to diagnose polygenic disorders like hypertension or diabetes. So why are we limited to monogenic disorders? Because we'll be using primers that target the specific site we wish to amplify. So you have to know where the mutation is. And then that amplified DNA can be sequenced to determine what exactly is the mutation, or we can compare its size or mobility with a normal reference control. So these are some of the monogenic disorders can be detected by PCR. A lot of them have been discussed in your CBL, like cystic fibrosis, Leishman, adenosine deaminase, PKU, OTCD, sickle cell, beta thalassemia. And early this morning, you discussed Fragile X, right? Premutation. It can be detected using PCR, as well as Huntington's disease. Well, for today's example, we use Huntington's disease as the representative monogenic disorder that can be detected using PCR. First, what are some of the facts about Huntington's disease? Huntington's disease is due to expansion mutation of CAG repeats in the Huntingtin gene, just like Fragile X is due to expansion mutation of trinucleotide repeats in the FMR1 gene. The mode of inheritance here is autosomal dominant, which means you only have to have one mutated copy of the gene to manifest these symptoms. This disease affects the basal nuclei or ganglia. If you remember in your anatomy, the basal nuclei is a mass of gray matter embedded on the white matter of the cerebral cortex, and it is responsible for modulating your motor movement. Thus, if there's a lesion affecting the basal nuclei, like in Huntington's, it will be characterized by excessive movement, such as chorea and athetosis. So how do we use PCR to detect? So let's say this is the normal variant of the Huntington gene with the violet part representing the CAG repeats, while this is the expanded mutated variant. So if we amplify this using PCR, at the end of the process, we have produced 1 billion copies of this DNA fragment. So these fragments are relatively short. While if we amplify this using PCR, the DNA fragments that will be generated are considerably longer. So as always, the process does not end with amplification of those target sequences. We have to determine the size of the DNA fragments or amplicons. This can be done through gel electrophoresis. After gel electrophoresis, we stain this and view this under UV light. We can determine now their size as well as their presence. If we see this, that means that the DNA fragments are large and therefore they have slower mobility. That indicates the presence of expansion compared to this. Let's have a hypothetical case here. J, 50 years old, developed symptoms of Huntington when he was 40 years old. Now his wife Mary is normal. They have 2 children, still very young, ages 10 and 7, and they're afraid that their children might have Huntington's disease. So you want to be sure. So everyone was subjected to PCR, amplification of the Huntingtin gene. Let's take a look at the case of Mary. She has 2 copies of Huntingtin gene, One is slightly longer than the other, but nonetheless, both are normal. In the case of J, one copy is considerably longer than the other one. This is the mutant variant. This is the one that causes Huntington's disease. So this is passed on to the child, that's bad news. While if this is passed on to the child, that's great. So in the case of Jessica, this copy of the Huntington gene corresponds to this. So this is the copy that comes from the father, and this copy comes from the mother. In the case of Martin, this copy comes from the mother, while this copy comes from the father. This is a copy with a expansion mutation. So Martin is only 7 years old, so there's no symptoms. The problem with Huntington is that it manifests in your mid-adult years. So eventually in the future, Martin will definitely develop Huntington's disease. So pwede nang paghandaan ngayon pa lang. So it does have a predictive value that allows you to prepare for that eventuality. We can also use PCR to detect organisms like virus. So this is the basis for the PCR that we use now for detecting COVID, but that is RT-PCR, but the principle is the same. So say this is the genomic DNA of a virus, and you have realized that this is the sequence that would be a good target for amplification because it is unique in the virus. Thus, the primers that you design should target this. So after isolating the DNA, it's subjected to PCR. So there will be denaturation, followed by primer annealing, and then primer extension. At the end of the 30 cycles, we have produced 1 billion copies of this particular DNA segment. Then we try to detect this particular amplicons using gel electrophoresis. If there's the presence of a band, it means we're successful in amplification. That means there's sufficient amount of virus that you can actually amplify them. If you have a negative result, that means the virus cannot be detected. There's not enough copy perhaps, or it's not even there. So that gives a negative result. Okay, so before we go on a break, here are some key points to remember. Restriction endonuclease are sequence-specific endonuclease in that they only recognize a certain palindrome sequence. DNA recombinant technology became possible because of the discovery of restriction endonuclease. DNA can be propagated by cell-based propagation or cloning that requires a host cell and a vector, with plasmid being the most basic of all vector, while PCR is an in vitro process that simulates DNA replication. What are the steps in PCR? It's a repeated cycle of denaturation, primer annealing, and primer extension. So let's have a short break, and then we'll continue with the second part. So let's have a 5-minute bathroom break and water break for everyone. See you after 5 minutes. Can you hear me? So for this part, we shall be discussing DNA sequencing, the blotting techniques, and the last one will be DNA microarray. Okay, so there are actually 2 methods for DNA sequencing. The older one is referred to as a chemical cleavage method. Now, there is no lab in the world that utilizes that method, so there's no point in studying that. Instead, we'll be discussing the other method that's still currently being utilized. This is called the Sanger's method, or sometimes referred to as a chain termination method. Just like PCR, it is also based on DNA polymerase reaction. So just like PCR, it also requires a DNA template, a primer, a DNA polymerase, but it doesn't have to be thermostable. DNTP is a substrate, and of course, a special ingredient. dideoxynucleotide, or sometimes we refer to it as the chain terminator. The conventional DNA sequencing was run on 4 separate reactions. Each reaction would contain the requirements, the template, the primer, the DNA polymerase, and of course, the DNTPs. And then each reaction would contain a small amount of the chain terminator, dideoxynucleotide. So that test tube 1, for example, would contain a dideoxy ATP, chain terminator for A. Another test tube has a chain terminator for G, and so on. What is this chain terminator anyway, and how does it compare with the regular substrate deoxynucleotide? So let's focus on the regular deoxynucleotide. What is being pointed out here is the 3' hydroxyl group, which is absent on the chain terminator. What do you remember about the 3' hydroxyl group that we keep on emphasizing because it seemed like the theme for our discussion last week? I know significance in 3' hydroxyl group. Anyone? Preparation for your
Absolute requirement for
Yes, because it's the target for the phosphodiester bond formation. If there is no 3' hydroxyl group, there is no target for phosphodiester bond formation. So if this is the one incorporated into the growing chain, it terminates further growth. Wala nang kakabitan. So what's the principle behind it? Most of the time, the DNA polymerase uses normal nucleotides and the DNA molecule grow normally. Occasionally, DNA polymerase uses a dideoxynucleotide, which terminates further growth in that molecule. So the random insertion of dideoxynucleotide leaves at least a few chains terminated at every occurrence. So what does that mean? We have here a simple illustration, animation of the process. So we have 4 separate reactions. And in this particular reaction, it contains all the requirements, such as the template, the primer, the enzyme, DNA polymerase, and the DNTPs that will be incorporated into the growing chain, as well as a chain terminator for A. That means there are 2 possible results for this one. If the normal A is the one incorporated, the process of polymerization continues. While if it is the chain terminator that has been incorporated, it terminates further growth. Like in this example, a terminator was incorporated into the growing chain and it terminates further growth. This leaves us with a fragment that ends with the letter A. Now, this mixture contains a lot of template primers. So on another template and primer, it's actually the normal A that was incorporated. So polymerization actually continues until another chain termination has been incorporated by chance. And yet on another template and primer, normal A was incorporated, which means polymerization continues until another chain terminator has been incorporated. So in the end, we have generated a lot of DNA fragments of different lengths, but all of them are ending with the letter A. These DNA fragments will be arranged according to size, from smallest to largest, using gel electrophoresis. I'm sure you know that this fragment corresponds to this band, and so on. So from here, we can now deduce the partial sequences of the DNA. So this is the smallest fragment. So we can say that at the 5' end, there is a presence of an A. Unknown, 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 unknown. We don't know how many unknowns there are, but we know there is another letter A. Unknown, unknown, unknown again. And we don't know how many unknowns there are, but we're sure there is another letter A. So now we know now the partial sequence of the DNA. How do we determine now the unknown parts in between? This is where the 3 other test tubes will come in. So like what I said, it requires a DNA template, a primer, and this is run on 4 separate reactions. The first test tube, all the test tubes actually will contain the requirements, DNTPs and DNA polymerase. One test tube will have a chain terminator for A, a G, a T, and a C. Let's go to the one with the chain terminator for A, like in our earlier example. Polymerization is taking place until a chain terminator has been incorporated, terminating further growth. So in the end, we have produced DNA fragments, all of which end with the letter A. Now, simultaneous reactions are also taking place in other test tubes, but they have a different terminator ending. So yung iba G, yung iba T and C. Now we have to figure out the sequence of the DNA. This is where gel electrophoresis will come in. So we can now deduce. The smallest fragment has a G followed by a T, an A, a T, an A, a G, a C, and so on. So you read a gel from the bottom going up. Sure, examination will be something like this. I would provide the result and you are going to give the sequence. So let's have a bit of practice. So the question is now, what is the sequence of the template? Anyone? What rule do we follow? How do we figure out the sequence of the template strand? Anyone from section B? Voila. Anyone? How do you figure out the sequence of the template? The answer is very simple. All right, I will not give the answer. I'll just let you figure that out. Okay, so nowadays sequencing is a totally automated process, but still based on Sanger's original technology. A laser beam scans the gel, places that information into a computer, and the computer prints out the result. Right, let's move on to the blotting technique. So this is a method to separate molecules in a complex mixture and identify the molecule of interest using a probe that has affinity to the molecule you are interested in finding. There are different types of southern blot, including southern, northern, western, and southwestern. So all of these blot are actually based on the original, which is southern blot. So they all utilize the same principle. So we use southern blot as the representative blotting technique for now. So the formal name of southern blot is DNA blot. It can be described as the analysis of DNA using a complementary radioactive DNA probe to locate this DNA. So it is a method to isolate and identify a DNA sequence of interest in a complex mixture. And you can do that as long as you have a probe that is complementary to the DNA sequence you're looking for. If you're wondering why it's called Southern blot, because it was developed by Dr. Edward Southern in the 70s. So it used to be the original workhorse for molecular biology before the development of PCR. How is Southern blot done? Just think of Cinderella. So Cinderella was actually found using a magical shoe that fits only her and her alone and not any other maiden. So we can liken Cinderella to the DNA that we are looking for in a complex mixture, while the magical shoe can be likened to the probe that has affinity only to Cinderella. So what are the steps in Southern blot? If PCR takes about 2 hours, 3 hours to complete, Southern blot is much more tedious. It takes about 4 days to complete the process. So day 1, the DNA is isolated and cut into smaller manageable fragments by restriction enzyme digestion. Day 2, the DNA fragments are arranged according to size using gel electrophoresis. Then the DNA is transferred from gel to paper by simple blotting to nitrocellulose paper. On the third day, the nitrocellulose paper impregnated with DNA is baked, denatured, and then hybridized with a probe. And the final step is X-ray photography to reveal the DNA we are interested in locating. To illustrate the process, let's say we have isolated genomic DNA, and this is the DNA sequence we are interested in locating. So this is our Cinderella. So our probe should be complementary to this. So the first step after isolating genomic DNA is to cut the DNA fragments into smaller, manageable fragments through restriction enzyme digestion. For the sake of simplicity, only 4 fragments are shown here, arranged according to size using gel electrophoresis. If we stop at this point, we are going to obtain a very neat banding pattern, but such is not the case that will occur in real situation, because with restriction enzyme digestion, you're going to obtain DNA fragments of every imaginable size, not just 4. So you're going to obtain a band here, 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 and everything in between. So instead of producing a banding pattern, what you produce is a smear. This would now obscure the DNA you are interested in finding. This is where the blotting becomes useful. A piece of nitrocellulose paper is placed on top. This allows the transfer of DNA from gel to paper. It's as simple as placing a tissue on your forehead, allowing the oil to transfer from your forehead to the tissue paper. Now we can do away with the gel. Now this paper impregnated with DNA is baked at high temperature. What is the purpose of baking? If we view it from the side, baking allows the DNA to cling permanently into the nitrocellulose paper. And at the same time, the DNA is denatured by high temperature. This would allow the DNA to accept its probe later on. So that DNA paper is taken out of the oven, placed in a solution that contains the probe. The probe will hybridize with all the sequences, but it can only hybridize with the one it is complementary with. So if we view it from the front, this is what we're going to see. Now, the final step is visualization, and this is through X-ray photography. Since the only one that cradles the probe, well, the probe is radioactive. So the only one that will be visible after X-ray photography is the DNA that cradles the probe. Thus, everything else will disappear except for this part. So this is the blot we are interested in finding. This is our Cinderella right here. This is the DNA we were looking for. So what are the applications of Southern blot? That includes gene discovery, mapping, evolution, development studies, diagnostic, and forensics. So it used to be the original workhorse, like what I said. But for today, we shall be discussing its application for diagnostics and forensics. For example, we can use Southern blot to detect point mutation in sickle cell. This version of Southern blot is called restriction fragment length polymorphism. Now, if you remember, in the original unmutated globin gene, one of the codons is GAG with point mutation, and now becomes GTG. GAG codes for glutamic acid. Now with a point mutation, it codes for valine. We can detect this point mutation. Let's start first with the normal. After isolating the DNA, it's cut using restriction endonuclease, generating DNA fragments. This is our Cinderella. This is one we are interested in. It's 1.2 kilobases in size. Our probe should bind to this. Now, if we subject the one with a point mutation to the same restriction endonuclease, it will not recognize this because of the change in sequence. So the DNA fragment that is going to be generated is considerably longer. So we next, as always, we arrange the DNA fragments according to size using gel electrophoresis. All other fragments no longer shown again. So same steps. We transfer the DNA from gel to paper. As always, bake to denature the DNA, then allow to hybridize with a probe. And the final step is X-ray photography to reveal the DNA of interest. So you can see here, its mobility and size is different from the normal variant, and thus we can see there is point mutation. Okay, so let's have a hypothetical case. So Jay here is 50 years old. He has sickle cell disease, even at a very young age. The wife is normal. And what about the children? So you ordered that everyone undergo Southern blot, and this is the result. Mary, all of her genes are normal, so she is homozygous for the normal variant, while Jay, all the globin genes have point mutation, so he is homozygous for the mutation. So for the children, this obviously comes from the father, and this comes from the mother. The same would be true for Jessica. So both children are heterozygous for the point mutation. So they might have some mild symptoms of sickle cell. Now we can also use Southern blot to detect full mutation in Fragile X. Have you had your discussion of Fragile X this morning? Yes or no?
Yes, sir.
Okay, that's great. So I hope you can understand what's going on here. So what are the facts about Fragile X? First, it's the second most common cause of mental retardation. What is the number one cause? Okay, good. So the mode of inheritance here is X-linked, but it's an anomalous X-linked pattern because it displays what we call anticipation. This anomalous pattern is dubbed as Sherman's paradox when they were not able to explain why. What is anticipation? Well, the symptoms become worse with succeeding generation. In other words, the grandfather is dumb, the father is dumber, the grandson is dumbest. That's anticipation. It turns out the Sherman's paradox, or the anticipation that comes out of it, is the result of expansion mutation of the FMR1 gene, the same mechanism that occurs in Fragile X. For some unknown reason, expansion mutation occurs only during oogenesis, but not during spermatogenesis. Okay, so how do we do Southern blot to detect Fragile X? So let's say this is the FMR1 gene with CGG expansion mutation, full version. While this is, say, for example, the normal variant. So we repeat the same steps as always. After isolating the genomic DNA, we cut it using a restriction endonuclease to generate fragments of manageable size, all other fragments no longer shown, and then arrange according to size using gel electrophoresis, blot to transfer the DNA from gel to paper, bake, no longer shown, and then allow to hybridize with a probe that binds to the CGG sequences. Then take an X-ray photograph to reveal the result. So you can see here, with full mutation, the DNA fragments are considerably larger compared to the normal variant. Okay, let's have here a hypothetical case. Si Jay na naman. Jay and Mary appear to be normal, but the family history of Mary from another side of the family has Fragile X. So they're scared that, you know, it could also run in their family. They do have two children, Martin and Jessica, now married, but they do have mild learning disability. And what about the grandchildren? They could have Fragile X. So you ordered everyone undergo Southern blot, and this is the result, right? So before we interpret the result, my first question is, how many, in what chromosome do we find the FMR1 gene? Anyone?
X chromosome.
Okay, good. So how many copies do men have of the X chromosome? Just one. So we only have one FMR1 gene for J. So this is normal. For Mary, she has two copies. Different sizes. One is normal, the other one is slightly long. What's the problem with this? It's in the pre-mutation range. What's the problem with this? copy? Anyone?
Possible kung ma-convert to full mutation.
Okay, so there's a possibility that during oogenesis, it can undergo expansion mutation and pass that expanded copy to the children. Let's take a look at the case of the two children, Martin and Jessica. Let's start with Martin. This copy comes from the mother or from the father? Anyone? Mother or father?
Father po.
Mother, is it from number 1 or is it from number 2?
From 1 po.
Number 1. Is it longer or shorter?
Longer po daw.
Okay, what's the explanation for that? Why is it longer? Simple, right? Ano explanation? Nagkaroon ng expansion mutation ito during oogenesis.
Since nga po siya kay mother, so may...
Okay, so that's it. Now, expansion mutation for some unknown reason doesn't happen during spermatogenesis. So when Martin passed this on to Sarah, it's stable. But fortunately, ito pre-mutation pa lang. Well, the same would be the case for Jessica. This copy is the same as this one, while this comes from the father. Now, what's the problem with this? Anyone? Anyone? Mahirap naman turuan ng section B. Pinakamahirap turuan sa lahat. Anyone? Kakaiyak. Anyone? Wala. Sarili kong tanong, sarili kong sagot. Jessica is female. She has oogenesis. So when this undergoes, this copy on the oocytes undergo oogenesis, they can expand, and thus when passed on to Brian, it is now the full expanded version. So it has a full mutation. So Brian has Fragile X. All right, so now let's discuss the other application of Southern blot, and that would be DNA fingerprinting. So we can analyze the restriction, digestion, fragmentation of a person's DNA. Now, the DNA sequences that are going to be analyzed here are referred to as VNTR, variable number of tandem repeats, sometimes called the microsatellites in other books. These are the non-coding regions in our DNA. Fingerprinting can be used for forensic and can also be used to determine your lineage. Why? The VNTR is unique for each individual, so it can be used for forensic, and it's handed down both by your mother and father, so it can be used for determining your lineage. So to illustrate, the part of DNA that is analyzed is called the VNTR. Well, obviously, because the sequences are in tandem, repeated several times, some are long, some are short, kaya variable number, a copy is comes from your father and the other copy comes from your mother. So our probe should bind to the VNTRs. So as always, after isolating genomic DNA, same steps, cut using restriction endonuclease. Generating fragments that will be arranged according to size using gel electrophoresis. Blot to transfer on a nitrocellulose paper, bake, no longer shown, and then hybridize with a probe that binds to VNTR. Take an X-ray photograph. You are going to obtain a banding pattern that would be unique for you, like a barcode. So since it's unique for you, it can be used for forensic purposes. Like say, for example, we have here the victim of sexual assault. There's the evidence that contains DNA from the victim and from the perpetrator of the crime. So there's the suspect 1 and 2, and then there's the boyfriend. Since the sperm DNA matches suspect number 2, you can tie in since the sperm DNA matches suspect number 1, you can tie in suspect number 1 to the crime scene. And thus, you can provide evidence that he could be responsible for the sexual assault. It can also be used to determine your paternity and maternity because it's a combination of your father and your mother's DNA. So in the case of daughter 1, you can see that this is the same as the mother. So it comes from the mother. This VNTR comes from the father, and from the mother. So you can say daughter number 1 is the biologic offspring of the mother. What about for the case of daughter number 2? Well, this matches the mother. This matches the mother. This doesn't match anyone, so it must have come from somewhere else. This matches the mother. This doesn't match anyone. It's slightly longer than this. So you can say that daughter number 2 is the biologic offspring of the mother from another man, not this father. Son number 1 is the same case as daughter number 1. While son number 2, the VNTR doesn't match anyone. So you can say son number 2 is not the biologic offspring of neither the mother nor the father. Let's go to the case we presented earlier, the case of Thomas Jefferson and Sally Hemings. So the kid Eston Hemings' descendants claim that Thomas Jefferson is their ancestor, so that the male descendants of Eston Hemings were subjected to DNA fingerprint, and they are going to be compared with the ideally male descendants of Thomas Jefferson. Bakit male descendants? The VNTR regions that will be analyzed here are those that are found only on the Y chromosome. So isang region lang ang titignan, Y chromosome lang. Why Y chromosome? Because it's handed down by father to son. Well, there's a complication to the story. Thomas Jefferson doesn't have a living male descendant. So yung uncle ni Thomas Jefferson, yung mga descendants ni uncle, ang gagamiting reference. Now, there's another complication to the story. There is the descendants of Thomas Woodson. They also claim that Thomas Jefferson is their ancestor. But the mother of Thomas Woodson is not known, hindi si Sally Hemings. So they were also subjected to the same test. And this is the result published in Nature, shown in numbers indicating the size and frequency of the NTRs. Since we're looking only at the Y chromosome, one locus, dapat perfect match. So if you take a look at Eston Hemings and the descendants of the uncle, it's a perfect match, pareho-pareho. That means they have the same Y chromosome, while the descendants of Thomas Woodson is totally different. So the Y chromosome here is different. So you can say that Eston Hemings and Thomas Jefferson have the same Y chromosome. He could have been fathered by Thomas Jefferson, or he could have been fathered by the uncle. Still a dispute even up to now. But what's definitive is that Thomas Woodson was never fathered by Thomas Jefferson, nor by the uncle. Okay, so like what I said, Southern blot is the original blotting technique, and there are offshoots such as Northern, Western, Southwestern. All of them utilize the same principle. What changes now is what is being analyzed. Like analysis of RNA is called Northern blot. The probe here is a radioactive DNA. Well, because DNA is very stable. Now, protein analysis is called the Western blot. The probe here is an antibody that has affinity for the probe. This is exemplified by the confirmatory test for HIV. The combination analysis of DNA, then we have a probe, an antibody that has affinity for DNA. This is called Southwestern. All right, finally, so there's DNA microarray. So this overcomes now the limitation of traditional biotechnology tools. And what is their limitation? They can only analyze a single gene at a time. Why is this a problem? Well, humans have about 30,000 genes. And most of the common diseases like hypertension, diabetes, heart disease, and so on are the result of mutation and interplay of several genes and not just one. So if you're going to use southern and northern blot to analyze those 1,000 genes simultaneously, well, one lifetime would not be enough. So you need to have a tool that can analyze them simultaneously. This is where microarray will come in. So this is somewhat like southern and northern blot, but done 1,000 times on a single DNA microchip. This microchip contains thousands of spots, and embedded on that spot are single-stranded DNA templates representing a particular gene. When they light up, that means that gene is active, and when they don't light up, that means the gene is inactive or has a mutation. What's the principle behind it? So let's say this is a simplified version. Embedded on each spot would be the DNA template representing a particular gene. To determine what genes are expressed, we obtain the RNA. Or RNA, of course, is very unstable, so it has to be converted first into cDNA by reverse transcription, and then label that cDNA with a fluorescent dye. They are allowed then to hybridize with the microchip, and they will hybridize with their respective template. A laser beam scans this. This will appear as a bright spot. This will appear as a dark spot. Well, the bright spot represents genes that are active, they are expressed, while the dark spot represents genes that are silent or have a mutation. So if you have a question, say, for example, what genes are active in cancer cells, you can easily narrow down those 30,000 genes into a few manageable ones. And if you ask, what are the ones that are inactive in a cancer cell, then you can get your answer using this. So what are the applications of microarray? You can identify gene mutation, determine the expression level of genes, determine SNP, single nucleotide polymorphism. It can also be used for drug discovery and toxicological research and disease diagnosis. All right, so let's just end the discussion with key points to remember. So DNA can be sequenced via Sanger's method, otherwise known as the chain termination method. And this is based on DNA polymerase reaction. So Southern blot is DNA analysis using radioactive DNA probe. What are its applications? This include detection of mutation and DNA fingerprinting. Now, if you wish to analyze 1,000 genes simultaneously, what you need to employ is DNA microarray. So this overcomes the limitation of traditional biotechnology tools. If you wish to read further on this topic, it is a chapter found in Harper's, in Lippincott, and Mark's Medical Biochemistry. You can choose the book that you feel comfortable reading. So that is the end for our lecture today. So do you have any questions? All right, good.